ladies and gentlemen. We come to our last presentation. Dr. Ellis has been waiting very patiently. We're going to talk about some modelling and methane analysis. Thank you.
Thanks. So thanks for hanging around for the last presentation. Hopefully it will be interesting. I kind of, as I was making this presentation, ended up retitling it as the 4 M's, which is methane, models, mitigation, and money, which I think is what it boils down to for me. When we look at feeding the planet of the future, I think we have an interesting set of conflicting demands, actually. On the one hand, we have this demand for increased production. So depending on who you're quoting, the global population is going to be over 9 billion, and the demand for meat and dairy product consumption is going to increase accordingly. At the same time, we have limited prospects to significantly increase the amount of arable agricultural land we have available, and therefore production has to intensify. Now, ruminants have a unique ability to turn human inedible feed into human edible food. And if we look at that on a, which one is the, oh, this one, yeah. If we look at this on a human edible protein basis, so we're looking at the ratio of the output of human edible protein in milk, or this example is milk or meat, over the input of human edible protein in the animal feed, we find that for ruminants, the ratio is generally greater than 1. It depends on which country you're looking at, and therefore the type of diet being fed. For example, in the US, the value is lower because we have a greater input of cereal grains in the diet, which are also edible by humans. Versus in the Netherlands, for example, they use a lot more grass and grass silage, so the value is a bit higher. And for example, in Kenya, the return is almost infinite because there's virtually no human edible protein in the diet of the animal. And if we compare this actually to, I kind of like the comparison, to swine or poultry production, their ratios are typically less than 1. And so ruminants do have an important niche kind of in the production of human edible protein. However, at the same time, ruminant production is also associated with significant greenhouse gas and nutrient excretion to the environment, which may be detrimental. So greenhouse gas production of concern are methane, nitrous oxide, and carbon dioxide, and nutrients that may affect soil quality and water quality are nitrogen and phosphorus. It's estimated that the environmental impact per unit milk and meat has to be cut in half by 2050 just to avoid increasing emission levels beyond present levels. So how much methane are ruminants actually producing? On an animal basis, a dairy cow is going to lose approximately 6.5% of her gross energy intake as methane, versus a feedlot beef steer may lose only 3%, and that's mainly dietary differences. Now globally, agriculture may contribute roughly 14.5% of the projected anthropogenic greenhouse gas effect, and roughly 40% of that is going to be from enteric fermentation via methane. So that's the entire agriculture sector total, and 40% of that is from enteric fermentation. And that's why methane is largely targeted as the source from agriculture that should be reduced. So if we step back and we look at the whole cow and we look at the conversion of feed into animal product, methane is one of several energy losses to the animal. So feed conversion efficiency for a dairy cow will be milk over feed intake, and methane is a part of that, but it's not the only part. So the other losses are, of course, losses in the feces due to incomplete digestibility of the diet, as well as in urine via nitrogen and amino acid breakdown, as well as the heat increment of feeding and maintenance energy expenditures. So inherently, we think methane and feed conversion efficiency are related, but what we're going to talk about today is whether that's always necessarily true. If we look at IPCC, the Intergovernmental Panel on Climate Change, suggests 3 tiers of how to estimate methane emissions from cattle. So the classic tier 1 approach is like this. There's a single factor. So methane is kilograms per head per year. So for example, in North America, it's 128, and Western Europe, it's 117. But we know already that methane is significantly related to the amount the animal is eating and the composition of that intake. So the tier 2 approach says, okay, methane is 6.5% of gross energy intake. And this will account for some of the variation in dietary differences between animals. But what I want to show is it doesn't always hold true. So IPCC suggests a third tier of methane emission prediction, and that's where it's actually more of a hypothetical tier, where countries are invited to develop their own kind of developments of the tier 2 approach or develop their own mechanistic models to describe methane emissions. As of right now, the Netherlands is the only country to have put forth such a mechanistic model to predict methane emissions. The underlying structure of that model is like this. So methane is produced as the result of reduction of carbon dioxide and hydrogen to methane and water. So methane and the hydrogen balance in the rumen are closely related. So hydrogen sources in the rumen are when we ferment carbohydrate and protein to acetate and butyrate. That results in hydrogen production. And also when amino acids when microbes use amino acids for growth in the rumen. Those are our main hydrogen sources. The big hydrogen sinks are when we ferment carbohydrate and protein to propionate and valerate, when microbes use non-protein nitrogen for growth, and also via biohydrogenation of unsaturated fatty acids. What I want to do today is actually go through kind of the top methane mitigation strategies that you see in the literature. And we're going to look at them from two different standpoints. We're going to look at them from a methane in grams per kilo of milk produced, and in terms of feed conversion efficiency. If we're talking about feeding the planet 50 years from now, where we want to both control our greenhouse gas emissions, but also feed a large global population, these are probably the two metrics that we need to look at, and we need to look at them together. Probably the biggest driver of enteric methane emissions is dry matter intake, and we see that very frequently, that as we increase dry matter intake on the x-axis, that methane, if we're expressing this as kilograms or grams per day and megajoules per day, it increases. So this relationship is not entirely linear, though. This is a bit nonlinear. So that's pretty straightforward. However, if we express this as methane as a percent of gross energy intake versus the same dry matter intake, we see a different pattern. So as intake increases, we tend to see that methane as a percentage of intake decreases. Now we can already see here a problem with using the standard IPCC Tier 2 6.5%. Now the reason for this decrease as intake increases is twofold. Number one, as we feed cows more, passage rate increases and therefore digestibility decreases. And number two, as intake increases and passage rate increase, you tend to have more propionate production in the rumen, which acts as a hydrogen sink. So you have these two mechanisms so that as intake increases, methane per megajoule of feed will decrease. Now, intake and milk yield often tend to increase together as we make cows more productive. So if we express methane per unit of milk versus milk yield, we see that as milk yield increases, methane relative to milk decreases. This is also for two reasons. Number one, the reason from the previous slide, where we have a decrease in methane per kilo of dry matter intake as intake increases. But also, as you increase milk yield, feed conversion efficiency tends to increase. And that's because earlier this morning they mentioned dilution of the maintenance energy effect. So if we are producing more milk per head, and if we look at the same total versus a lower milk yield, then loss to maintenance is a lower proportion of that total with less animals. So in general, this one's pretty maybe generally accepted. As we increase intake, we can see reductions in both methane per kilo of milk and also improvements in feed conversion efficiency. Now, we can talk at the end that there may be some upper limits to this mode of action. Probably the second most important driver of methane emissions in cattle is carbohydrate source of the diet. So if we go back to this model, we know that fermentation of some carbohydrates will lead to acetate and butyrate, and some others will lead to propionate and valerate. And so the balance between these two will largely determine or contribute largely to the methane emissions. If we look at some modeling work that's based on in vivo data, For example, Hendrickson developed this equation where they looked at kind of the relative contribution of different components of the diet, but this is on a digested basis. So this is fermented organic matter. So we get the most methane from sugar, from fermented sugar, followed by cellulose, hemicellulose, and starch. In a separate modeling work, Bannock also produced a similar ranking of carbohydrate sources, where this is relative methane formation, and it's relative to sugar. So we can see the relative ranking of cell wall starch and protein, but also that this relationship is dependent on pH, so it depends on a lot of other factors that are going on in the rumen as well. This is a big slide, and I'm going to kind of explain it to you in detail the first time because I'm going to show it to you again. This was some modeling work we did a couple years ago where we wanted to simulate changes in forage composition. So this was ryegrass, and looking at how methane emissions and milk yield and digestibility would respond to changing the composition of that grass. So what we did was anywhere you see an LN, that's for a low nitrogen fertilized grass, and HN is a high nitrogen fertilized grass. And then we looked at increasing the sugar or the water-soluble carbohydrate fraction of that grass at the expense of crude protein, NDF, or a mixture of both. And then we looked at 3 exchange levels, so either 20, 60, or 90 grams per kilo change. So if we compare, for example, this is a basal simulation, and in this one in red, we increased the sugar content at the expense of NDF. What we saw was a response in milk yield, and this is due mainly to an increase in the organic matter digestibility of the diet when you increase the sugar content. So that's good. Because we held dry matter intake constant and we saw a response in milk, we saw an improvement in feed conversion efficiency. And we saw a slight improvement in methane per kilo, expressed per kilo of milk. Now, methane in megajoules per day was actually predicted to increase, but because of the simultaneous increase in milk yield, the net effect was a reduction. So overall, this one gets an interchanging arrow because carbohydrate composition is a large and varying component. But we do definitely have options here to improve both methane per unit of milk and feed conversion efficiency at the same time. Now, forage quality and carbohydrate source are largely interrelated. Forage quality we're kind of defining as a combination of chemical composition and digestibility at the same time. This is some preliminary results from colleagues of mine at Wageningen, where they looked at improving forage quality via 2 mechanisms. And the first one was cutting the grass earlier, so cutting it at 3 weeks instead of 5 weeks. And the second one was applying 2 different rates of nitrogen fertilizer to the grass. What they saw with cutting the grass earlier was a response in milk. So this is fat and protein corrected milk yield. And actually, everything in my presentation where we talk about milk is fat and protein corrected. So they saw an increase in milk yield with the cutting earlier, therefore an increase in feed conversion efficiency, and also a reduction in methane per unit of milk. So that's good. When they applied nitrogen fertilizer, so they also saw an improvement in an improvement in milk yield, an improvement in feed conversion efficiency, and also a reduction in methane per unit of milk. However, we can talk about a bit later that this strategy may have implications for other nutrient excretion, such as nitrogen, and we'll come to that in the end. We simulated similar things within our mechanistic model. So this is comparing a low nitrogen simulated grass to a high nitrogen fertilized grass. So similarly, we could simulate a response in milk yield via an increase in digestibility of the diet, a decrease in methane per unit of milk, and also in feed conversion efficiency. So definitely, if we work at increasing forage quality, we can improve both methane per unit of milk and feed conversion efficiency at the same time. The next kind of 4 ones I want to talk about are all via kind of similar mode of action, which is these can all act on the hydrogen balance and act as alternative hydrogen sinks within the rumen. So when we feed fat, if we go back to this slide too, We know that the biohydrogenation of unsaturated fatty acids is a hydrogen sink. And so some of the reduction in methane we see or expect to see with fat are because of this. But also, when we increase fat in the diet at the expense of fermentable organic matter, we're decreasing the amount of fermentable organic matter in the diet. So we see a general reduction in fermentation end products as a result. In vivo, what this looks like, for example, this is an experiment looking at the addition of linseed via different forms, so crude linseed, extruded, or linseed oil. And this is methane per kilo of dry matter. We see a general reduction, so they saw more than 50% reduction in methane per unit of dry matter intake via feeding linseed oil. However, they also saw a decrease in milk yield, and this was because of a decrease in organic matter digestibility. They also saw no change in feed conversion efficiency. So if we talk about fat, then yes, we can reduce methane per unit of milk, but perhaps we can't improve feed conversion efficiency. Nitrate is an interesting one. Nitrate is a very strong alternative hydrogen sink for the rumen. So the reduction of nitrate to nitrite and then nitrate to ammonia is very energetically favorable in the rumen. So it will outcompete the reduction of carbon dioxide to methane or even sulfur to hydrogen sulfate. However, there's a bit of a star next to this one, of course. There's a risk of toxicity with this additive. The reduction of nitrate to nitrate is very fast, but the reduction of nitrite to ammonia requires some adaptation period. It's not as quick to happen. And so if you have an accumulation of nitrite, it can be absorbed into the blood and turn blood hemoglobin from a ferrous to a ferric form, which can generally render it unable to carry oxygen. So there's kind of a star next to this one. But we do see when animals are adapted to it, reductions in methane per unit of dry matter intake. They've seen no changes in milk yield and a reduction in methane per kilo of milk. They also saw no changes, however, in digestibility or no changes in feed conversion efficiency. Now, within, for example, the IPCC Tier 1 and Tier 2 models, we can't represent such a mitigation strategy, and this is one of the downfalls of simplistic models. However, we also have not represented it within a mechanistic model yet, but it would not be difficult to add it to such a model as an alternative hydrogen sink. So nitrate is another one where perhaps we can see reductions in methane per unit milk, but maybe not necessarily in feed conversion efficiency. Ionophores, such as monensin. Now, these are known to or used to improve feed conversion efficiency via an inhibition of gram-positive bacteria in the rumen. And a few years ago, we wanted to be able to incorporate this into a mechanistic model for beef cattle to quantify we talk about this as the long-term effect, perhaps, of monensin as opposed to a short-term response. So we wanted to quantify the change in VFA profile in the rumen. So we saw significant increases in the propionate and butyrate and a non-significant decrease in acetate. But essentially, we can develop equations like this to account for feed additives like this within a mechanistic model. And so essentially, those equations are applied to this VFA stoichiometry. And in vivo, we see, as expected, some improvements in feed conversion efficiency with monensin feeding, and also some reductions in methane per unit of milk yield. So ionophores, maybe not in being applied in every country. However, we do see the potential here for some improvements in both methane and feed conversion efficiency. But I should say that the caveat for kind of all of these ones at the bottom are that, for obvious reasons, a dose is important and also basal diet in terms of the amount of response you're going to see. So I kind of generalize here, but there are, of course, exceptions to all of these. The last one that I want to talk about is a relatively recent potential methane mitigator, which is 3-nitroxypropanol. This is a synthetic compound, and it inhibits methyl coenzyme M reductase. So that's the final step in reducing carbon dioxide to methane. In this recent study from 2014, they actually saw no change in intake or milk yield. some non-significant improvements, which resulted in a slight but significant improvement in feed conversion efficiency, and very drastic reductions in methane. So methane per kilo of milk was reduced by almost 60%, which is quite impressive. I should add that other studies from 2014 on this feed additive have not produced such dramatic results. The study by Reynolds and colleagues did not see such drastic reductions, although they bolused it directly into the rumen. The interesting thing about this one is that they also measured, for example, the acetate to propionate ratio in the rumen, and the change in acetate to propionate ratio could not account for the metabolic fate of hydrogen. So because of this, we have an excess of hydrogen that can no longer go to methane anymore. And it is apparently not being accounted for by acetate and butyrate. So that's kind of a question. And in the study from Reynolds, they saw decreases in digestibility of the diet. And so overall, I think that this is an interesting one, but certainly needs more research done to understand the mode of action. So again, we can't account for this in any of our current models used for inventories, but certainly it could be added. It can be added. So feed additive, I put a question mark beside it. It definitely has promise, but perhaps we need to know a bit more about mode of action. So we're kind of getting close to the end there. Where are we in terms of modeling methane emissions? Well, I hope that what you've seen is that enteric methane can vary substantially based on diet, and that these two tiers of approach really can't account for that variation. And if we're talking about applying mitigation strategies and being able to generate national budgets, what we really need is some better equations. I did this really rough calculation where I looked at different equations. I looked at the tier 1, the tier 2, a mechanistic model, and I threw a couple of my own in to criticize my own equations as well. We can generally, this is just this was from the same forage composition simulation database that I showed you earlier, and I just looked at the minimum, maximum minus the minimum methane predictions. So that gives you kind of a range, or how variable the equation is able to predict the responses. So, for example, we see no variation from IPCC, minimal variation from some of these simple equations, more variation being able to account for in the more complex equations, but by far, a mechanistic model can explain a lot more of what's going on. And this is probably even less than what's in reality. The other point I think that needs to be made is methane the whole story? And of course, the answer is no. We did some simulations where we looked at methane per unit of milk versus nitrogen utilization ratio, and what you tend to see, unfortunately, is that as something becomes more advantageous for methane, it tends to be less advantageous, for example, nitrogen excretion. There may be some exceptions to this, so there are some little data sets in here where they were beneficial to both, but you do tend to run into nutrient swapping a lot with these two, and therefore, what we need to do is move towards more whole farm modeling of nutrient excretion if we really want to understand what's happening to greenhouse gas emissions on farm. So these are some of the whole farm models that are kind of out there. As we go from single greenhouse gas to multiple greenhouse gases and multiple nutrients, we do find that the equations used within them tend to get more simple. So what we really need to do is focus on kind of getting up here. So to conclude, what I think the focus for the future should really be on reducing methane emission intensity while maintaining or improving feed conversion efficiency. There are multiple mitigation options available, but also units are important, and I often wonder whether how IPCC represents their emission factors should change to be per unit of product to make them a bit more relevant. And of course, the movement towards more complex mechanistic or semi-mechanistic models is going to allow us to generate more accurate emission estimates, but that we also need to consider greenhouse gases as a group to assess environmental impact. And I'd like to thank my collaborators and also for Biomin for inviting me, and might have gone over a bit, but thank you. I'll take any questions you might have.
Thank you, Dr. Ellis. Any questions from the floor? Please, in the middle. Got a few minutes.
Yeah, okay.
Thank you. What's your idea about essential oils? Because you didn't mention it.
Yeah, I of course didn't address all mitigation options that are available. There's certainly been a lot of research done on essential oils, or curiosity in it, for sure. I think we can definitely, with some of them, see reductions in methane, but I think we may run into some of this trade-off. Like, I'm not sure that you would see improvements in feed conversion efficiency at the same time. You may sometimes be associated with drops in digestibility or milk yield. So it seems like, in general, that if you can improve feed conversion efficiency, you probably are going to improve methane. But if you improve methane, you may not always improve feed conversion efficiency. So it's kind of an interesting balance there.
On the lipid supplementation, you showed mainly data on linseed oil, which is the main source of unsaturated fatty acid. Is there any data out there showing reduction of methane when feeding saturated fatty acids?
Yeah, if you feed saturated fatty acids, so of course you won't necessarily have the biohydrogenation sink, but what you will have is, when you're comparing it to a control diet, a reduction in methane, because you're decreasing the amount of fermentable organic matter in the diet by introducing fat. So you will see a reduction, but it's via the exchange of organic matter for or fermentable organic matter for infermentable. Yeah.
Thank you very much for a nice presentation, and indeed a very thorough overview of the mitigation strategies. So I was just wondering, do you include in your modeling long-term effects of those feeding strategies? So in terms of including feed additives, but also feed components that you use, that you mentioned in the presentation, let's say concentrate amount, because concentrate amount might be that at the short term might decrease the methane mitigation and improve the efficiency, but probably in the long term might have some negative impact on health and increase the unproductive days of the cows.
Yeah, when we, at least for the mechanistic modeling I talk about here, we're running the model to a steady state. So we're not looking at a short-term, within, like, for example, within a day, we're not looking at a short-term response. What we're really trying to represent is like the average long-term response. Of course, like, it can become challenging with certain additives. For example, trying to model monensin, for example. With monensin, short-term responses tend to be quite significant, but it was always questioned whether that's a persisting effect. What we did when we modeled monensin, for example, we tried to just look at the effect on VFA profile. So monensin can also have an effect on protozoa in the rumen, and that can have the short-term effect. But when we modeled it, we tried to model just the VFA profile response. So trying to capture kind of some of the longer-term impact. And so when we simulated it, we saw smaller reductions in methane than you may see in some of the short-term in vivo trials and stuff like that. But I don't know if that answers your question, actually. But yeah.
Yes, certainly, but because in order to give a more holistic view, because the long term of the modeling is to provide a model how to use in the practice, let's say.
How to use a model like that in practice.
Exactly.
Yeah, so for example, what the Dutch are doing is they have such a mechanistic model as their tier 3 approach. And what they're working on right now is creating a user interface so that it would be available to feed companies, farmers, producers, whoever would like to utilize it, because actually using such a mechanistic model can be quite complex and maybe not necessarily available to everybody. So I think the key for using more advanced models like that is developing interfaces that have boundaries also on inputs, so we can't make the model blow up.
Okay, everyone would like to join me again in thanking Dr. Ellis for her presentation.